Hello and welcome to the Microbiome Medics podcast. I'm Dr. Siobhan McCormack.
And I'm Dr. Sheena Fraser, and we're your co-hosts.
We are both GPs and lifestyle medics with a shared passion for microbiome science. We'll be translating the evidence and packaging it into actionable, bite-sized chunks so that you can harness the power of the microbiome to improve your own health and that of your patients. So hello and a very warm welcome. I'm Dr. Siobhan McCormack and today's episode is dedicated to fibre, the Cinderella nutrient. We're going to deep dive into why this overlooked food component might just be one of the most powerful tools you have for your metabolic, immune and gut health. Well, if fibre is the Cinderella nutrient, then my guest today must be the fairy godmother. Joining me is the fibre-fabulous Dr. Emily Leeming, PhD, microbiome scientist at King's College London, registered dietitian, chef, presenter, and author of the best-selling Genius Gut, the life-changing science of eating for your second brain. Emily, welcome.
So great being here. Thanks so much for having me.
So, Emily, before diving into fantastic fibre, can you set the scene by explaining how you came to become a microbiome researcher from sort of sixth form to now? What happened?
Oh, wow. I would say I've definitely not had the most conventional career. I went and worked as a chef on superyachts, traveling the world after my undergraduate in nutrition. I had no idea what I wanted to do. I felt completely lost. And I've always had an absolute love of food, love of cooking. And I, would go off and do kind of short-term freelance things from, I guess, 16, 17. This is what happens when you grow up in the countryside. There's not many actual proper full-time jobs to do in that, that you want to do in the holidays during, school holidays and university holidays. So for me, that was actually, funnily enough, at the time, obviously, you're going to get told, when are you going to get a real serious job? You're swanning around the oceans, traveling from different island to island. But I really feel that was so seismic in terms of understanding and appreciating food, how people eat, how to cook, and also that kind of work ethic. I think if anybody's worked in hospitality, you have to just give it your all. And I think that's really been a philosophy that's brought me through. So kind of fast forward, I then moved to Australia and did my master's in dietetics to train as a dietitian. Never thought that I was going to be heading towards academia. I thought I was going to be maybe setting up my clinic, which I did for a while. And I read a book, actually it was Gut by Julia Enders, who's this German gastroenterologist. That was my first insight into what the microbiome even was, that we had this like fascinating collection of trillions of tiny living organisms that are living in our gut and influencing our health. And it just took me on this journey of, I went and did a research internship at King's to see whether I was kind of interested in research. I got a research scholarship when I went back, for my master's, because that was kind of during a holiday break. And it just went from there. And I then went and did my PhD in the gut microbiome, looking at kind of how diet influences the gut microbiome, but also how can we look at diet in new and novel ways to understand that relationship better. And then I still now work at King's, but also kind of maybe a full circle back to that book, Gut by Julia Enders, in the sense that it was, a popular science book that sparked my imagination, and now kind of being able to do that with my own writing and my own books, and feeling very strongly, like you do, I know, about making science accessible and interesting and engaging, and really kind of bringing it to the reader so everybody feels in charge of their own health.
Oh, that's brilliant to hear your story. I love, different ways people have come into microbiome science, because it is a relatively new science. I love that book, Gut. They had those, we'll put it in the show notes because it really is worth looking at. It really spoke to me as well. So it's got these lovely little pictures that were drawn by her sister, and it really communicates the ideas well. And as you say, they were very new at that time. And the other book that I liked was the Psychobiotic Revolution, which was kind of Ted Dinnan and Cryan book. And again, that was my wow, we've got an Amazonian rainforest in our gut. I need to go and tell everyone. Why isn't everyone talking about this? So yeah, I do get that. The way it's communicated is hugely important. But before we deep dive into dietary fibre, I just wanted to set the scene a bit for the listeners, because I think we can forget that fibre's made by plants for plants. So it's a very important part of the cell wall of all parts of the plant. So stem, leaf, seed, fruit, stalks. There's loads of different types of fibre, and I think that's important for people to understand. We don't necessarily have to know all the different types, but it's very important for the architecture of different types of the plant that you have all these different components, different types of fibre with different functions. So some give rigidity and strength, some are flexible. You've got fibre that's important for storage, that's important for the growth of the plant, water retention, to fight against predators as well, and even seed dispersal. So I kind of think that's a really important place to start, because plants make fibre and we steal it. We use it in textiles, cotton and hemp. We use it in our coir and sisal carpets, in bamboo socks and paper. But we also eat it. And as a medical student in the 1980s, we had about one line on fibre. It was really boring. It helped constipation. Otherwise, it's irrelevant. And the only other memory I have in the 1980s was my mum, who's always on a diet. And we had this around the place was this Audrey Eaton's F-plan diet. So my mum would sort of be miserably eating like bran flakes. So all seemed a bit dull and miserable. So how have we gone from that to fibre is now the coolest kid in town? What's happened? And is it all about microbiome science?
I think we've got a lot to thank for microbiome science bringing fibre forefront in the last couple of years. And I would say that's because fibre is effectively a kind of favoured fuel source for your gut microbes. They feed on certain types of fibre, as you say. It's not all these different types of fibres that we have. Certain types of fibres feed your gut microbes, and that's what then helps them to kind of make these molecules for your health, and particularly these short-chain fatty acids. So not just helping with the health of your gut, but also these short-chain fatty acids can travel widely across your body and even as far-reaching as your brain. And this is kind of part of the reason why we get so excited about your gut microbiome. It's not necessarily kind of who's there. It's what are they doing? What are they doing for our health? So I think there is an element where, yes, it's microbiome science, but I think it's also, I think it's really interesting in nutrition. We do see these kind of shifts of waves of interest in different nutrients at different times. I think we're coming off the back of a protein craze, not to say that protein isn't important, but I think we've become so obsessed with protein. It's been at the detriment of all these other nutrients, including fibre. And when we think about the stats that 96% of us in the UK are not reaching the recommended 30 grams of fibre a day, that is, a pretty significant, population shortfall. But in a way, I kind of love to spin that to say that, okay, well, actually, it's kind of great to know that most people, have this one thing that maybe we can then double down and look to fix. So I would say there's been, off the back of the protein craze, yes, this new microbiome science, which is really signalling this new role that fibre's playing. But again, fibre for decades now has been one of the most researched nutrients that has, really strong, consistent evidence to show that it's helpful in lowering risk of heart disease, type 2 diabetes, bowel cancer. But I think it's really had this very boring, dry kind of bad PR. It's been just something, it's seen as something that just was, bland, brown and stodgy that just helped you do a nice poo, which obviously it's great. I'm always thrilled if someone tells me they do a good, nice bowel movement. I'm like, that's fantastic. But I think the other thing is, as you say, it's been associated with, the F plan of kind of this is going to just help you fill up and kind of bulk up your stomach, which it does as well. But I think, circling back to what you said about all these different types of fibres, and it's just amazing to kind of break down actually how these different types of fibres can have different characteristics, and these characteristics can also overlap. So some of them can be feeding your gut microbes. You call them the kind of fermentable or fermenting fermented by your gut microbes. We have other ones, yes, we've got that traditional soluble versus insoluble, which we're now slightly shifting away from as being just a bit too basic. But that effectively is saying that if it's insoluble, it's going to add a lot more bulk to the food that you're eating. That's going to help to stretch your stomach, to help send those signals back to your brain that you feel full. It's also going to add bulk to your poo and help things move through in a timely manner through your gut. So you're going to have less contact of potential harmful compounds with your gut wall. And then on the flip side, we have soluble, where it's going to dissolve in water. And it is fascinating to think that some fibers you might not even be able to see because it's completely dissolved in water. And that's very kind of counterintuitive to what we expect fiber to be, as this kind of thing that we're going to chew on and munch on. And then within those soluble fibers, you've got viscous fibers, which are effectively fibers that kind of can thicken and maybe sometimes form a gel. And these then have their own kind of set actions in your body, which are, helping to lower levels of cholesterol, which is part of the reason why we see this kind of really strong relationship between fiber and heart disease.
So I'm getting the idea that fiber's just not one, it's an umbrella term. And under that umbrella, we're finding, as science progresses, more and more complexity as to different composition, different types of fibre and the health benefits of different things. I mean, what you said there about not expecting, we expect it. I've already said you make coir and sisal carpets out of it and material and cotton. So it does give that idea that, it's all like straw. And yet there are some types of fibre that and some foods that you wouldn't expect to contain fibre and are very, very different. I mean, give me some examples of some foods which people are not expecting to be fibrous, but have got plenty of fibre in them.
I would say avocados are usually a surprising one, because we associate it with being this like creamy, kind of gorgeously delicious, kind of rich, fatty flavour. And that's actually one of the kind of higher fibre vegetables, if you don't argue with me and call it fruit. Whereas on the flip side, we've got salad leaves, which, I think most people would associate as being high in fibre, because you've got that process of chowing down and having to chew on something, and it feels, you can feel the texture of the leaves. And actually, that's pretty low in fibre in comparison to other vegetables. It's only about 2 grams of fibre per 100 grams. So that's like a very large bowl of just lettuce leaves. So in a way, you can actually be thinking that you're eating, yes, you're having like wonderful lots of different types of nutrients if you're having lots of salads, but you might not still be meeting your fibre requirements. And I think we've really got this, what I really want to kind of bring onto people's plates, if kind of excuse the pun, is just, we know when we talk about protein that, most people can say off the top of their heads what top protein sources are. They say, fish, meat, eggs. I really want to bring that home for fibre and do fibre justice and kind of shift away from this, okay, fibre is just having to chow down on salad leaves or kind of a kind of misconception, or if it's just kind of brown, stodgy, brown bread, all bran, that really kind of heavy wedge of food. And actually kind of circle back and be like, as you say, like it's the key component of, plants, of all plants, sort of plant structures. And you can find it in fruits, vegetables, beans, legumes, nuts, seeds, whole grains. And actually, if you're going to make things really easy for yourself, it's really like doubling down on what food groups are giving us the most bang for our buck when it comes to fibre. And actually, fruits and vegetables, fantastic. We do need to eat more of them. But they aren't actually providing us with nearly as much fibre as you would get from whole grains, nuts and seeds, and beans. And yet we're really not eating that much of either of those three. Only, I think it's less than 18% of people in the UK aren't eating any whole grains at all. And I think nuts, on average, it's like 3 or 6 grams a day or something, which is like teeny tiny amount. So that, we're talking about easy, simple swaps here, like Can you choose a whole grain pasta, whole wheat pasta, instead of your usual white pasta? Tastes pretty much the same. Can you have maybe a handful of nuts and seeds with your cup of tea? Keep the jar by the kettle so it can signal or remind you that when you're making your cup of tea, I'm going to have a handful of nuts. Small, simple things. And again, beans, I think the beans have had a moment. Maybe that's also part of the reason why we've got fibre on the rise, because I think beans have also had their culinary heyday in the last year or two.
Yeah, absolutely. Yes, there's a few kind of a lot of good Instagram feeds on beans. Incredibly affordable, very nutritious, and in today's climate of financial issues, I think families are always looking for those affordable bulk cooking options. But Emily, before, I really want to spend most of the time actually talking about practical aspects. But just before we get there, there's quite a lot of healthcare professionals who listen to this podcast who may be a bit not convinced how important fibre is. I've got a few friends who are really keen on the protein discussion, and that's their kind of key nutrient. How do we know? How can we convince people with the evidence, and what types of evidence are there? I know that your background is epidemiology, so I wonder if you could just give us a flavour of some of the evidence that's available. How can we be confident that fibre is so important?
We've got decades of research to really show how even just small increases of fibre, so I'm talking about 7 grams of fibre, for example, that's the same as half a can of beans. Just adding that even if you're not meeting the recommended 30 grams of fibre a day, is related with a significantly lower risk of heart disease, type 2 diabetes, and colorectal cancer, so bowel cancer. I think most of the evidence that we have does actually, interestingly, tend to centre around heart disease, and that's really where the recommendations of that 30 grams comes from. That 30 grams is what's associated, what we see from kind of population studies and patterns, that the people who are eating the kind of 20 to 30 grams have that lower risk than the people who are eating the least amount. What's really interesting there is we don't actually really have that much consistent evidence for kind of higher amounts of than 30 grams. So there is this kind of thinking that the benefit it will continue. So really it's something that we're thinking, yes, this is reach the 30 grams, but above that could also give us significant benefit as well. And that was kind of, those recommendations were put in place probably like 10 years ago. And then we've got this new kind of coming into really understanding with the microbiome, really helping to support the microbiome, providing that fuel source. And what we're seeing from for example, studies in mice, that if mice are not getting enough fibre, so they're able to look within mice to see what the impact on the gut might be if there's not enough fibre around. And they see that actually that kind of mucus that protects the lining of your gut ends up then becoming some kind of backup food source when there's no fibre around for the microbes to feed on. That can then break down some of that mucus, meaning then those microbes and potential kind of harmful substances can then reach the gut wall. And I think particularly like thinking of stepping back to in terms of just thinking about kind of gut health in general, in terms of not just the microbes, but the home that the microbes live in. And we have, this rising rates of bowel cancer in people under 50, where it's traditionally been this disease of people in their 70s. And of course, there's a lot of unknown factors there. Not to say it's purely diet. There's lots of other environmental lifestyle factors that could be at play. But one key thing, and this is something that we're researching at King's at the moment, is thinking, is it because we've got this lack of fibre, this relationship with the gut microbiome, but also that kind of transit time through the gut and that contact with the gut wall. So a lot of consistent evidence, and I would say, very well recognised as one of the nutrients with the most consistent evidence to support it in terms of health when you're eating it in enough quantities.
Yeah, and that reminds me, we can't have an hour-long conversation about fibre without mentioning Dennis Burkett who was a kind of surgeon, epidemiologist in the 1960s, worked in Uganda, I think, who was looking at the differential non-communicable disease rates between people who ate rural African food and then people who were eating Western food. Discussed various diseases, including diverticular, colorectal cancer, and heart cardiovascular disease as well. But it was interesting. He was talking about the rural Africans having something like 100 grams of fibre a day. So although we're failing to reach our 30 grams, there are populations out there who are on much higher levels. And are you saying that would give us even further protection, or we don't know whether there's, increasing protection the more fibre you have?
So there are some of the epidemiology studies where they've seen this trend, this quite linear trend between upping your fibre and having a kind of corresponding increase in health benefits. Some of the studies the larger population studies have then modelled and seen that relationship seems to continue up until 40, 50. Then we have, a handful of kind of small studies, some of it quite old, that have looked at that higher quantity. So, for example, there's the portfolio study, which effectively puts people on different, very kind of high fibre, high soluble fibre, because they were very interested in this kind of cholesterol response and heart disease. And I think one of the groups was in this very, very high fibre, kind of high nuts, high fruit. And we're having kind of around, I think, 140 grams or something across 2 weeks, which, of course, is, not something that is feasible for the everyday person. I think it was equivalent of like 5 kilos of food a day, which is, a mammoth amount. And so we have got some, and showing that was, I think, like a 30% kind of reduction in LDL cholesterol or something, because they were trying to pull in all the evidence that we know around foods and cholesterol levels. And so we have got little kind of hints and insights of these higher levels. But I think the key thing here is that we don't want to, I think there's two things. So first off, we don't want to end up in the mess that we've ended up with protein, where protein is important. We do need to care about protein, but not to the lack of everything else around it. And I think there's this kind of, a worry again that with these really kind of high amounts, that we then don't end up having a balanced plate. And then the second thing is that feasibility really is that we know that it can take your gut time to adapt. Your gut, feeding your gut fibre, for your microbes is a little bit like throwing, your microbes Glastonbury when it hasn't, had any parties ever in its life before. That can then, it's not causing any harm, but it might then give you some gut symptoms like bloating, perhaps constipation, some kind of pain. So what we really try and say is, if you are going to up your fibre, up it low, start low, go slowly, so across weeks, and make sure that you're drinking plenty of water as well, because some types of fibre, again, can act a bit like a sponge and need water to kind of work their best. And I think because we don't have that really strong evidence for anything really, I would say 30 plus, we're in a good zone. 40, 50 will be really interesting. And again, that's something that one of the aspects we're looking at in the research study that we're doing. We're looking at 40 grams versus 30 grams, because we're quite interested to see if there's anything that we can see that is significantly different there. But in terms of like pushing up into above those levels, although there has been, ancestral diets that of course have hit kind of 75, 100 grams, different population groups, that it just seems we just don't know yet if that's really possible. Whether if it is possible, do we need to be there? Are we going to see the strongest benefits? And actually what we do see at the moment in terms of the biggest benefits is simply moving from a low fibre diet to that 30 grams, or even just shifting that 7 grams above. So just I think it can feel a lot of the time, the nutrition, when you have a kind of threshold to meet, that if you're not meeting it, then you're not getting any benefit. But it's those small changes that are consistent over time that's really going to have the effect on your health, even if it is simply an extra 7 grams a day and you're not eating a 30 grams.
So those people out there who are eating very little fibre, they're going to get benefit from just increasing a tiny amount and start low, go slow, and gradually build it up. So I think that's really good advice. Emily, if we can focus in, you've mentioned colorectal cancer. There will be some people listening who aren't aware of this big concern there is globally about the increased rates of colorectal cancer in younger people. I wonder if you can give us some context to that and then go on and explain a few of the mechanisms by which fibre might be making a difference.
Yeah, so this is something that we are kind of collectively looking into. It's a kind of multi-institution research project that's led by Harvard. And so one of the kind of research aspects that we're in charge of at King's is what we're calling the kind of Eat Fibre study. And this is really kind of coming from a place of seeing that there's differences in how bowel cancer is presenting in young people versus in old people. So it's kind of quite more frequently a different part of the colon. It is also presenting sometimes a bit more aggressively. So there's different. It also seems to have less of a kind of heritable aspect to it. So you're less likely to inherit it from your parents. In terms of obviously that just being a small number of people. And so there's different factors that seem at play here. And I think we're collectively looking at this from different angles across the different institutions that are involved. Coming at this, obviously, as a dietitian and microbiome, we're interested in that kind of diet aspect. And really kind of, I think in nutrition, we're quite often kind of a little bit skewed to just looking at things that we think are harmful. We know that, for example, excessive red meat consumption and processed meat consumption is related to kind of a higher rate of bowel cancer. But for us, it was like, can we understand this protective effect of fibre that we see in bowel cancer for people who are, older generation? But can we understand a bit more about, is it how much should we be having more of it? But also this kind of newer question of really understanding and bringing in what you were saying about different types of fibres being really important. And so we're really looking at this like fibre variety aspect as well. We're giving set recommendations for beans, for whole grains, for nuts, and really to kind of try and encourage having a variety, but also bringing in those foods that tend to give the biggest kind of fibre oomph. And that could then make it easier to reach that 40 grams a day. So very much through the lens of, can we design the study in a way that we hope will give very simple, practical advice that clinicians can take to their patients? A key thing with fibre is really just making sure that you're not like the transit time, I think, is a really key part of it in terms of the relationship with bowel cancer. So that's really how fast is food moving through your gut, going from your mouth all the way out the other end. And we want that to move at a timely pace so that anything that might be potentially harmful is going to have less time in contact with the gut wall. But it's also that relationship with the gut microbiome as well, in terms of what molecules are they producing, how is that impacting the health of the gut and the corresponding environment. And then also like this really interesting insight into that kind of, types of microbes. So there's certain microbes that we see that, like, for example, fuso, which is effectively a very common bacteria that's found in your mouth, absolutely normal. Everybody has it. It's completely plentiful, but we don't tend to see it in the gut unless you see it in people with bowel cancer. And is it that these changes in the gut microbiome, are they happening as a consequence of that already becoming a kind of cancerous environment? Or is it that these microbes and this pattern, this signature of different types of microbes, it's not just that type, are also kind of having a kind of mechanistic effect they're causing something to happen, and they might be kind of a kind of key player, and then therefore can we change that through the intervention that we're doing? Can we see a shift that is beneficial?
Yeah.
This podcast is funded by the British Society of Lifestyle Medicine. The BSLM is an independent charity set up to promote the role of lifestyle medicine in improving people's health and well-being. We empower healthcare professionals to support patients to prevent, manage, and even reverse many chronic conditions through education and connection to our like-minded community. Shiv and I are both members of the BSLM and certified lifestyle medicine practitioners. We authored our first course, The Fundamentals of Gut Microbiome Science, for BSLM. This podcast was created with the aim of improving education on the role of human microbiomes. For those keen on doing a deeper dive, please check out the course on the BSLM Learning Academy page. Listeners to this podcast can access a 10% discount with code MBM10. We're also able to offer all healthcare professional listeners 50% off the first year of membership, unlocking a free overview course and nearly £100 off annual conference tickets, using the code MICROBM in capitals. Now let's get back to the show.
Oh, that's so interesting. It sort of reminds me of one of my favourite sort of studies, the O'Keefe 2015 study with African Americans. So people may know that compared to rural African Americans, people who then move to America in this case and become urbanised, westernised diet have far higher rates of colorectal cancer. And it was this lovely study which did a food swap. So these rural African people were subjected to westernised foods, so this kind of ultra-processed food, for only 2 weeks, I think, and vice versa. The African Americans who were now living in America had rural African diets. And the incredible thing was, the changes were seen very rapidly, and it was only 2 weeks. I don't know how they persuaded them to do biopsies. I'm pretty sure they did biopsies as well. But one of the things was, I think, the bioelastic signature was markedly changed. I've got a feeling that the rural Africans, none of them had any polyps, and the African Americans, I think about 30 or 40% of them did, and which is a sort of prerequisite for colorectal cancer, I understand, although please do correct me. But it was amazing how quickly changes. I suppose that's one of the exciting things about the science, is that, changes seem to happen very quickly. Is that something that your study is finding, or how are you looking at this?
Yeah, so we're looking at, we're collecting, so we're not going to be collecting microbiome measures. We just kicked off the study now, so you're getting it as it's literally in real time. So obviously microbiome samples are incredibly expensive. We're also taking lots of different other measures. So we are very fortunate in the sense that we are collecting a real wide remit of different measures, from your poo, from your blood, from your pee. So that is going to be really interesting. But I think it's also for us really this feasibility lens as well. So we are actually collecting on a regular basis gut symptom questionnaires, because I think a big part of this is that seems to put off people from fibre. We can talk about the power of fibre until the cows come home. But I think a lot of people feel very nervous about eating fibre because they might have suddenly eaten, a whole meal, a big bean chili, and suddenly said, they've suddenly eaten, 15, 18 grams of fibre in one sitting, having not usually had it, and having lots of different symptoms come up. And also, talking about things like farts, for example. We see from some prebiotic studies where they've given them certain doses of prebiotics that it takes a few weeks for their gut to actually just adapt and settle back down to the regular kind of gas output. And so we see this kind of microbial shift in getting excited. They make gas as a side effect when they feed on fibre. That is a great thing. I think we've got to give a little bit of gas is fine. But I think little things like that, where it's, yes, we're having, we can see, great benefits. And different studies have shown different things on this, but we know that it tends to be with diet, that it's long-term benefits that, most of the studies tend to be kind of 3 months long, and we see kind of significant changes within that. But there could be, within the first, 6 weeks or something. Usually I'm sure there's likely a benefit there as well. But just seeing this also, that recognizing that any gut symptoms are likely to kind of settle back down. I think that's also really important from a kind of clinical perspective of just kind of putting people's mind at ease, that if they are feeling a little bit more bloated or have got a bit more gas, that your gut then is, should be able to, for most people, kind of adapt back to baseline. And for us, it's kind of being able to hone down when that might be, but also different people will likely have different responses, and can we then understand that a bit better? So it might be that, a certain type of person, it takes a little longer, and can we then give certain different advice for that?
Yeah, you'll be pleased to know we did a whole hour on farting, destigmatizing farting. So, if you want to listen to a whole hour, then there's a podcast episode on it. But I just wanted to stop there for a moment, because I think nutrition and nutrition science gets a hard time. People are sort of saying, oh, they keep changing their mind. Why can't they decide? Why don't they know? 1 minute it's this, 1 minute it's that. I just want to step back a bit. And if you can just give us an idea, you've got a research project sort of, going off at the moment. Can you give people some idea of the complexities of the planning, the collaboration, the cost in terms of people power, the time, the, all the different specialities that are involved, and why it's so difficult to do nutrition studies?
Yeah, I mean, I think there's two things there. I feel that firstly, it seems like nutrition science is changing all the time, because our kind of, the news sources that we're getting, and this is, coming from someone who, writes a newspaper, and I can kind of see this from the other side, is that we are drawn to headlines that are going to cause us to click. That's how these things work. And I think there is this kind of lens that we then see nutrition through, where, no journalist really, apart from, us kind of scientists and clinicians, really want to talk about fruits and vegetables, because it's not new and exciting. So it seems like advice is constantly changing. Should you eat a banana or not eat a banana? Whereas actually that advice has stayed pretty much, I would say on most fronts, has stayed pretty stable over kind of years and decades. It's just how we kind of getting and accessing that information. How is it being fed to us? So I think that's a kind of key thing I want to bring up. The second thing, yes, it is very time consuming, and there is a lot of aspects to think about when it comes to researching nutrition. And I think that hasn't helped. One thing that I think hasn't helped in that sense, in terms of nutrition seeming confusing, is that it's a science that a lot of other researchers who don't work in nutrition research are quite happy to tack on, a diet, gathering some diet information, and then kind of interpreting that without the expert lens of having kind of worked in that area. And so therefore you can end up with something that potentially, but they haven't adjusted for this, or they haven't looked at it in this way. And it can then be incredibly confusing. And I think there's, I'm quite, would somebody who was a meteorologist start kind of collecting soil samples and thinking they can interpret soil samples? No. Maybe that's not the right, I need to come up with a good comparison there, but I think you get the idea. Yeah. If you were, a tiler of a roof, would you think that you can talk about piping? Not necessarily. So I think there is this kind of very interesting lens of, I think because we all eat, we think we know food. With nutrition studies, the biggest difficulty that we have is really actually getting people to stick to the diet. That's the thing that we talk about a lot. Because you can, if we all, and we know this through, if we all kind of have a pretty good idea of what healthy eating is, for example, yet it is very difficult for us to stick to it, because there's this behavioural, it's kind of science element where it's behavioural change is very hard. And it's the same with running any kind of diet intervention. It's getting people to stick to the diet is incredibly hard. But there's a lot of thinking about, okay, what resources, what touch points with a dietitian. And then even just before that, there's a huge planning stage in terms of kind of planning the study, obviously getting the funding because they're incredibly expensive. We think even just collecting the samples, getting the samples then processed throughout the study, and getting that kind of data analysed. And then we've got a whole team of researchers who are, working on this for years. There's a lot of different elements to consider and to factor in. And even just, the selection of your participants as well, because you don't want to throw the cat amongst the pigeons and have, for example, people taking antibiotics just before, you're taking their microbiome sample, and making sure that all these different factors are considered so that the data that you end up collecting is the best quality that's available, has all the factors that are considered. And therefore, you can then analyse it and hopefully come up with some findings that are useful. And, for me, it's very much a practicality element, but that's not always the purpose of research. It doesn't always have to be. And again, for us, it's also trying to understand some kind of mechanistic part as well, because I think with microbiome science, we've seen a lot of patterns in populations in humans, and we've seen the mechanisms in mice, but we're now shifting to, can we see this relationship that we see in mice? Can we see this in humans? And how can we look at this in a way when we're not collecting biopsies, for example, of people's guts, because that's very invasive and very expensive? Are there certain markers that we can use to try and understand this relationship, for example, with fibre, with the gut barrier lining, with the microbes, with the molecules they're producing, and, with the gut barrier itself as well, is this becoming more, permeable because there's a lack of fibre there? And then are we seeing a shift in that by the end of the intervention?
So, how long did it take you? Your studies just kicked off for you. Has it been a year of planning or more than that, or, in total?
Yeah, it was. So we've had the team grew, I would say, 8 months prior to the study kicking off. I think I was involved from a year and a half prior. So yeah, it's been, that's. So we've had different people kind of come on board throughout. So we have our kind of study lead who's, there full time. And we have the different PIs. We're also running a community project as well.
PIs are what?
Principal investigators.
Okay, yeah.
It's kind of like managers.
Yeah.
The study dietitian. I mean, there's a lot of little different factors to work. And then that's also working. We're very fortunate in our department to already have a cohort of 14,000 twins that kind of take part in our research projects. And within the department, we already have a kind of operations team. So it's kind of a lot of collaboration within the department, and also, discussions with the potential participants or participant representations, representatives themselves. Do they feel that that this is something that they would do? Is there anything that they think they would feel uncomfortable with? And again, even embedded in our, the bigger, wider research project for bowel cancer, we have people with lived experience of bowel cancer who are, very embedded throughout the institutions, different projects and different institutions, because it's so important that we have this connection with people who've got a lived experience of bowel cancer. I think there's really learning from the fact that in research, we don't always discuss or talk to people who actually have the disease themselves and what their priorities are and what they wish could be found. What could science be supporting with? So that's Rena, I think, sadly, I think quite a new experience for me to have that so easily accessible, but something that we're really finding incredibly impactful, and thinking of different ways that we can also interact with our kind of patient advocates, and can we kind of, think about when we're writing our research papers that we're doing some kind of public translation piece that can go with it, so that we're connecting with people by through how we're doing that research, and because it's too often that these research papers just get locked behind a paywall, and no one has access to them unless you have, a university affiliation where you can kind of get these papers.
Yeah, I like this idea of co-production and research and how much richer it makes it. And that's really exciting. But I think, again, just for listeners, there's all this ethics and consent and all these incredible complexities. And yes, needing the participants to, follow the protocol, because otherwise it's all, all for nothing. So how many participants have you got in this study?
We have 100 twin pairs. So we've got one twin on one diet and the other twin on the other diet. And we ask them not to talk to each other too much. And that just means the beauty of twins is that this study wouldn't have to be on twins, but we're just fortunate because we've got this cohort. It just takes some of that kind of genetic factor out. So we know that it's kind of anything change that we're seeing is likely more related to the diet, that environmental change. And it's great. We're incredibly fortunate to have, really, really fantastic. These twins are so enthusiastic and love being part of the research, and we're incredibly grateful to have them, because without them, we wouldn't be able to run the study. And I think the other thing that we have, which we're really looking forward to, is the fact that we've had relationships with many of these twins for a really long time, and they come in and have their clinic visit, because we are really an epidemiology department by trade. So we've kind of collected their data over time. So also being able to then look through the lens of, if we see, a certain group that we want to kind of dive into, we've got their microbiome samples from kind of years prior. So yeah, that's also something that's really valuable.
Yeah, this Twin UK cohort comes up again and again. It's just, they must be quite amazing group of people, actually. It's sort of certainly hard working in the research world. Just talking about that, the numbers in a study. So, lots of papers I've read in nutrition studies, there can be 10 people or 20 people or 60 people. And then you've got this new perspective or new paradigm in studies where you've got these massive data sets, and that's, for example, the PREDICT studies. Can you just give me, or just give us a quick, it's a possible question, but an idea of the PREDICT studies and how groundbreaking they are in terms of the sort of number and the perspective which you look at dietary sort of data from?
Yeah, so I was involved with the PREDICT studies at the kind of very beginning. So not now, but it was a really exciting time to be involved. So this was a collaboration. So Sarah Berry, who's a professor at King's College London, really kind of steers and kind of is at the forefront of leading these this PREDICT research. And it's I think the first thing that's really struck me particularly at the time was the power of this collaboration between what is possible between industry and academia. And of course, there needs to be guardrails on that relationship in terms of, in general, in terms of how that happens. But for this, effectively meant that you were able to collect, multiple different samples on a really wide, population, but a very deep, granular detail that we necessarily wouldn't, be able to collect unless you had millions and millions and millions of pounds in an academic research setting. And I think also the pace of which that data was then accessible. Absolute kind of wealth of research. So it meant kind of being able to collect. Yeah, I think, this is, again, with microbiome research, for example, just to kind of use that as an example, you really need to have a good group, we're talking about 100, 200, participants for that to be kind of meaningful. You might see something that's a kind of signal, but you really need to have that power in numbers for your results to actually kind of say something. And that for most people, in terms of specifically kind of, 5-10 years ago, where the sequencing costs were astronomical. I mean, of course, this is what happens. You have a new technology gets brought in, but then of course that then becomes a bit more kind of more affordable because it's being used a lot more often. So those rates of the costs have now gone down, but it is still expensive to do. So then to have this huge amount of data, not just of the microbiome, but also looking at diet in a really kind of granular way, looking at postprandial responses, so looking at blood sugar responses and blood fat, and particularly kind of triglyceride responses, in kind of the kind of 6 hours prior, 6 hours post a kind of fat challenge within a muffin was fascinating, and I think really signals how can we learn about this in the future? How can we kind of tie together and use apps and technologies, whether it's a kind of collaboration with, a bowel cancer charity or kind of a long COVID kind of group? How can we, pull people together? And I think probably maybe that missing piece again is the patient advocates, have the lived person experience with those diseases and have, that huge phenomenal ability to have cohort growth, and what could that potentially signal in terms of what we find. So yes, but I think the key, yes, we were able to do a lot more. They were going to do a lot more structured in terms of the research at the beginning, but then it ends up being that we still have a lot of value in very kind of closely tuned randomized control trials as well, which I think it's just adding another picture, another kind of color to the tapestry of how we can understand science.
Yeah, so one of the spin-offs was your blue poo study, which you were co-author on. I wonder if you could just give us a quick outline of that. And then my question is, should I be looking at my poo? Does my transit time matter? Should we be looking at our poo every day? What are your thoughts? Are there cultural differences in what people do around the globe?
So the blue poo study was effectively the participants were fed some bright blue muffins that looked like something off Sesame, the Cookie Monster on Sesame Street. They were kind of, quite radioactive blue. And what we asked the participants was to kind of measure the time from them eating it to that poo appearing blue at the other end. And that is effectively what we look at when we're talking about transit time. So how quickly is that food moving through your gut from start to finish? Traditionally, the best gold standard approach of doing that is you kind of swallow a little kind of pill, and it's very kind of precise measure. But again, that's expensive and it's not always possible. So this was a kind of, can we use this approach as a way of kind of understanding gut transit time? And I think also really playing on that understanding that the gut microbiome is incredibly complex. There's lots of different factors that are influencing it. So when we come to research, we want to know what those different factors are so that we can account for it. And so a kind of big rationale for the study was really that actually how influential or how related is the gut microbiome to how quickly food is moving through your gut. And we were able to see pretty kind of strong relationships between the gut and the kind of who's there for kind of slow transit time. So anything that's kind of taking a couple of days to go through. And then we had this kind of normal transit time, which is all trying to think in terms of clinical parameters. And then this kind of speedy transit time, which was, I think, less than 14 hours or something. And seeing that we could see that there was this strong relationship with the gut microbiome, which kind of shows that we should probably be taking some measures and taking it into account when we're doing our analysis. I think one interesting thing as well was kind of that we kind of recognize that I think, microbiome diversity in the sense of having lots of different types of microbes is our kind of best but very imperfect measure of, quote unquote, what a healthy microbiome is. But in a way is also incredibly flawed. And I think a kind of great example is with, constipation and this kind of slow transit time, that actually you can have this proliferation of different types of microbes because they're getting longer time on that food source, but it's not necessarily, quote unquote, healthier. And actually what might be happening is it's also then shifting in terms of their food source from that kind of carbohydrate fiber to feeding more on protein, any kind of leftover protein that hasn't been digested in the small intestine. And that could then signal, we tend to see with those kind of diets, that if there's a kind of heavy animal plant-based diet, that the molecules that the microbes are making tend to have to be kind of less healthful and could potentially be harmful. So there's a kind of interesting kind of insight there. But effectively it was showing that, yes, this is not going to, we didn't look at it against the gold standard approach, but we were able to see a stronger relationship using this kind of measure of the blue poo than if we just looked at kind of poo shape and poo frequency, which is quite often what gets used as a proxy instead. So it was a bit of a fun but nerdy kind of methods paper in terms of what might be useful.
It is interesting that something as simple as transit time, or even, you talked about the Bristol stool scale, may have some utility in that they give some information about the bowel function, which, together with other information can be very useful. And also, just I take your point that this idea that increased diversity is always a good thing. There'll be lots of nuances to that. And it's very, very sort of, we need to know a lot more. I watched your popular TED talk on fibre as the missing nutrient, and I loved the quote, "Feeling great is not about cutting foods out, it's about adding foods in instead." And I think one thing I really like about you, Emily, is you're really careful about your messaging. You're very, it's all about abundance and joy and enjoying your food. And I suppose in your communication, I think, you're unique in that you're, trained as a cook and a scientist as well. And that's an unusual combination. You care about how food looks and tastes and the joy of eating. Tell us a little bit about your book, why you wrote it, what it was like to write, and what the main messages are.
So Genius Gut is effectively a kind of overview of the latest science of the gut-brain connection. So how does your gut talk to your brain, but also, how is your brain influencing your gut and your gut microbiome? And then very kind of much, what can you do about supporting your gut microbiome through the food that you eat? And also really from a kind of whole body perspective. So I think it's just a kind of fascinating exploration into what is really a very young emerging science, this gut-brain connection. But yet at the same time kind of rooted in our everyday language, because we talk about gut feelings and gut instincts, having butterflies in our stomach. So we've kind of had this kind of conversation in terms of the gut-brain connection. So really, I think came from a deep love and interest of wanting to kind of share where we're at in terms of the research, but in a way that is, I hope, kind of still very nuanced, because I think it's, again, an area that can be completely taken off into the hills and kind of, people saying that, for example, serotonin is all made by your microbes. And actually, the reality is a little bit different in the sense that, yes, your microbes are involved in your serotonin production, but that tends to be the serotonin that's in your gut that can't cross your blood-brain barrier. But actually, your microbes, do seem to be involved in helping to supply those building bricks that are needed for serotonin in your brain that are then small enough to make their way through your blood-brain barrier. So kind of little interesting kind of nuggets and quirks there, but then very much, a big, as you say, kind of thing that I'm very keen on is this practical element. So yes, here's all the exciting science, but what can you do about it? And what do we know about the research around food and mood? Really, fascinating kind of landmark study from Felice Jacka, who I know we've both spent time with, and, really kind of kickstarting what has been, a lot of randomized control trials since. I think that was
2017, yeah. I mean, amazing. It was the first randomized controlled trial of its sort on mood and food. I mean, that's pretty astonishing, isn't it, considering, on sort of Mediterranean diet. But, I mean, I read your book a couple of weeks ago, and Emily, I really enjoyed it. I recommend it to interested public, but healthcare professionals. I learnt loads from it. It's, beautifully referenced. But the main brilliant thing about it, I could have stopped halfway through and thought, that is a great book. But then there was another half, which was all about, it's your culinary expertise and your dietitian's expertise and your microbiome scientist's expertise all in one, because you've just got these really actionable points. You've got these charts. You've got, what does it look like? What does a handful of fibre look like? Where do you get it from? The idea that chocolate, it's really difficult for my head to get into my head that, dark chocolate contains fibre, this thing you were talking about right at the beginning. It's absolutely brilliant the way you've done that. And as I said, I read it on my Kindle. I'm going to have to buy a hard copy because there's so many great charts that are really practical for people at the supermarket. Just to give people a flavour, if you're talking about just a breakfast, a realistic one-day sample menu from the book about something that might be easily done, affordable for breakfast that will get you up to a certain amount of fibre, just give us a couple of examples.
Yeah, so I would say if you're a sweet breakfast person or you've got a bit of a sweet tooth, porridge or overnight oats is a really great place to start. Oats, fantastic source of fibre. They also contain beta-glucans, which are also got kind of cholesterol-lowering properties. And so add into that, I really enjoy stirring through kind of nuts and seeds. So that could be that you're going from one end of the spectrum, that might be chia seeds and flax seeds, or it might be simply just having a handful of almonds or pumpkin seeds on top. Have that with some higher fibre fruits. So some examples of that might be pears. Any kind of berry usually tends to be high in fibre. The reason for that is because they contain little seeds in them, and seeds are pretty high in fibre. So any kind of seedy fruit, like passion fruit, for example, is going to be on that higher end of the fibre spectrum when it comes to fruit. And then if you're kind of thinking, again, always through the lens of, what is the balance there? So having some Greek yogurt with that, or some skiers, also high in protein, is just going to give you that kind of balanced roster of about 10 grams of fibre. And I would say that would push you to about 25 grams of protein.
So that takes us to you've just brought out one book. You've now got another one coming out. What's happening and what can this add to what I think is the perfect book?
So this is taking the practicalities to the next level. So I've got my next book, Fibre Power, which is effectively your guide and your 4-week plan to eating 30 grams of fibre. This is a real stepwise approach to help really recognising that needs to be that low and slow method, and drinking enough water. And there's lots of different tips and tricks that I give you along the way in terms of what fibre is, but also how to increase it in a way that feels enjoyable and satisfying and great for your health. And then there's 60 kind of colourful recipes in there as well, kind of tapping into my background as a chef on Superyachts, and kind of real full circle moment for me. But very much, again, that real kind of practicality, what is useful. I felt very strongly that most of us don't always have the energy to open up and do a full recipe every single day. So I've got what I've called food combos, which is kind of like if you chucked these few things into a pot, whichever way you wanted to do, just these 5 ingredients, you will get your protein and you'll get your fibre.
Wow.
So just like, sometimes, if it's not even, it might even just be like a sweet potato with, chicken breast and some green veggies. And it's like, okay, but that's not a meal, that's not a full recipe. But I know that by eating those things, and maybe I'm adding in, some cherry tomatoes, okay, that is going to give me what I need.
That sounds wonderful. Listen, have you got Microbiome Medics podcast listeners? Could we have a bean recipe? Is there something you could send us and we could put in the show notes? Just kind of, because I think it's, and when's that book coming out? When can we get our hands on it?
That's coming out in May, so very soon, May the 21st.
Listen, Emily, this has been a wonderful tour of fantastic fibre, and from short-chain fatty acids to blood pressure to immune, metabolic health to culinary expertise. It's been absolutely fantastic. We'll put in the show notes where people can find you online and all the details about your new book. I could talk to you for forever, but maybe you'll come back sometime because there's so much more to talk about. And we'd love to hear the results of your study on colorectal cancer in younger people when the results come out. But thank you so much for your expertise and for your time. Thank you, Emily.
Thank you. Thanks so much for having me. It's been great. Always love chatting with you.
Thank you so much for listening to this episode of Microbiome Medics podcast. We really hope you enjoyed the content and we welcome your feedback. We'd love to hear any suggestions you might have for microbiome topics that you'd like us to cover. And we also appreciate listeners' questions and will endeavour to answer them in the next podcast.